Welcome to Lecture 8.1, in which we will go over prokaryotic cell division. Our learning objectives for this video are to understand how the prokaryotic genome is replicated and describe how prokaryotes divide through the process of binary fission. Ultimately, our goal is to compare and contrast binary fission with mitosis, which we will learn about in the next video, and meiosis, which will be the next lecture. As the cell theory states, all cells come from pre-existing cells. New cells are formed by cellular reproduction, which involves a cell splitting itself up to form two new cells. When cells reproduce, they also need to duplicate their DNA so that the two new cells each have their own copy of DNA. The genome refers to all the genes and DNA in the cell that need to be duplicated. This is a series of images of a cell dividing over time. The cell body is the hazy gray area in the background, and the DNA is in the brighter color in the center. This shows the DNA condensing throughout cell division and being split between two cells. Recall that the prokaryotic genome is contained in a single circular double-stranded DNA found in the nucleoid region. Some bacteria also have smaller sets of DNA in the form of plasmids, but the essential genes are in the nucleoid. Replication of prokaryotic DNA is relatively simple. It starts at the origin of replication, which, as its name suggests, is where the DNA first opens up and replication begins. After the origin opens up, replication occurs bidirectionally, which means that the two strands of DNA are replicated at the same time, but in opposite directions. This results in two copies of the genome that can now be divided between two cells. We will learn more about DNA replication, particularly in eukaryotes, in a few lectures. Binary fission is the process of dividing a prokaryotic cell into two cells. Fission means to break apart, and binary refers to how the process of breaking apart forms two complete cells. We can see in this GIF how two E. coli bacteria divide into 4, then 8, then 16, and so on. Mitochondria and chloroplasts also divide this way. Since prokaryotes don't have a nucleus, DNA replication can occur along with cell division, making the process much simpler and quicker than eukaryotic cell division. In fact, some bacteria can divide every 20 minutes. Binary fission can be divided into 5 steps. In step 1, DNA replication begins at the origin of replication. There are some green dots labeled as FTSZ proteins floating around. These become important in step 2, in which the cell begins to elongate, and these proteins begin migrating to the center. FTSZ proteins are similar to tubulin in eukaryotes, which we will see is important for cell division. Ultimately, the FTSZ proteins help with physically dividing the cell. In step 3, the replicated DNA begins to separate and move towards the opposite ends of the two forming cells. The FTSZ proteins form a ring, and in step 4, the FTSZ proteins form a structure called a septum. The septum is essentially a partition or a wall that physically divides the cell. Finally, in step 5, the FTSZ proteins constrict and help pinch the cell into two separate cells. Ultimately, binary fission can be summarized as a process of DNA replication, cell elongation, DNA separation, septum formation, and cell division. In this video, we learned that DNA replication begins at a single origin of replication in prokaryotes and continues bidirectionally. We also learned that prokaryotes divide by binary fission, which involves replication of DNA, elongation of the cell, separation of DNA, and formation of the septum by FTSZ proteins to physically separate the cell into two. In the next video, we will learn more about eukaryotic chromosomes and cell division. Thanks for watching.